Hello students, welcome to Biosmart Academy YouTube channel. In today's lecture, we will continue our amino acid lecture series. Okay, previously we were uploaded 2 lectures on amino acids. Okay, in the first lecture we mainly discuss about the basics of amino acid, and in the second lecture we mainly deal with the classification of the amino acid. I hope that you all watched that lecture, and that 2 lectures are very important to understand today's lecture. Okay, in today's lecture we were going to discuss about the structures of the amino acid. Each and every structures we will discuss in details about the amino acids with single letter word. Okay, because structures and single letter words are very important from examination point of view. Okay, so this will be the part 3 of the lecture series of amino acid. In today's lecture, we will mainly discuss about the structures of amino acid with single letter word. Of each and every amino acid. Okay. So, specifically in today's lecture, we mainly deal with the non-polar amino acid structures. Okay. So in today's lecture, we specifically deal with the non-polar amino acid structures, and in the next lecture, we will discuss about the polar amino acid structures. Okay, as we classified amino acid into polar as well as non-polar. Okay, so let's start today's lecture, that is the structures of amino acid, that is non-polar. Okay, so just put the heading non-polar amino acids structures. Okay, non-polar amino acids structures. Clear? So the first non-polar amino acid is glycine. Okay, so glycine. Okay, glycine. So its single letter word is G. Okay, its single letter word is G. G for glycine. So its single letter word is also G. Okay, remember this. So what will be its structure? First of all, remember one point, that glycine is the simplest amino acid. Okay, glycine is the, some important point regarding glycine. Okay, it is the simplest amino acid. Okay, it is the simplest amino acid. This is the first point. Okay, and we discuss some point after discussing the structure. So let's look at the structure of glycine. So this is a structure of glycine, that is C alpha. This is acid group. This is amino group. This is hydrogen atom. Okay, and we draw the side chain with yellow. This is a side chain. So in glycine, the side chain is basically hydrogen, that is similar to this hydrogen. As a result, the side chain and this hydrogen atom are both the same. It means what? It means this carbon does not have the 4 arms different. That's why glycine is the only amino acid. Glycine is the only amino acid among 20 amino acid which is basically achiral in nature. And as a result, it is optically inactive. Okay, clear? So this is about the glycine. Okay, and one more important thing about the glycine is that glycine majorly present in turns. Especially basically proline and glycine majorly present in turns. Okay, proline and glycine majorly present in turns. Okay, and one more important point about the glycine is that if glycine is present in alpha helix structure, so this is alpha helix. Suppose here glycine is basically present, then it will disrupt the structure of alpha helix. Why it will disrupt the structure of alpha helix? Why it is so? So we will discuss all these things when we will learn the secondary structures of protein. Okay, so just remember these points about the glycine. Okay, proline also disrupt the structure of alpha helix. Okay, so we will detailing all these things in this secondary structure of protein when I will teach you secondary structure of protein. Okay, Clear? So this is all about the glycine. Okay, now look at the next amino acid, which is basically non-polar in nature, and that is alanine. Okay, that is alanine. Alanine. Okay, so what is the single letter word for alanine? The single letter word for alanine is A. Okay, A for alanine, the single letter word is also A. Just remember like this. Okay, clear? Now structure of alanine. So in alanine, this is a structure C alpha, this is the common, these three are common in all amino acid structures. Okay, now we draw the side chain with yellow. So the structure of alanine is in side chain CH3 group is present. So this is the side chain. Okay, clear? So you can see in these structures that carbon have 4 different hands: hydrogen, CH3, that is side chain, CO minus and NH3 plus. So carbon have 4 different hands. As a result, alanine is chiral in nature. Okay, as it is so chirality, so it is optically active amino acid. Clear? Okay, now note one important point about the alanine is that alanine basically it is the most favorable, it is the most favorable amino acid to form secondary structures, Okay, why it is the most favorable amino acid to form secondary structures? We will discuss all these things when we will discuss the secondary structures of protein. Okay, just note this point now. It is the most favorable amino acid that form secondary structures. Okay, so this is all about the alanine. Clear? Now next amino acid is valine. Okay, so the single letter word for valine is V. That is V for valine and V for single letter word is valine. Just remember like this. Okay, clear? Now just look at the structure of valine. See, C alpha, CO minus, hydrogen and NH3 plus. These three are common. Okay, and remember one point, draw the structure with me. Okay, so that you get memorize the structure. Okay, so we know that in alanine CH3 group is present. Okay, but in case of valine, just remove the 3 and put CH and just make 2 CH3 like this. Clear? This is the structure of valine. Okay, that in case of alanine we see that here 3 is present. So in valine we just remove this 3 and just put CH and just put CH3 here and here. So this is the structure of valine. This is the side chain of valine. Clear? Okay, so valine is also optically active and chiral in nature and chirality, show chirality. Clear? So this is about the valine. Okay, now let's look at the structure of the leucine. Okay, now let's look at the structure of leucine. Okay, so L for leucine, single letter word is also L. Okay, now just look at the structure of the leucine. These three are common, as you know. Okay, NH3 plus, this is the hydrogen atom. Okay, now we draw it with yellow. So in valine we see CH and in both the hands of CH that CH3. So in case of leucine, just put extra CH2 and all the structure is same like that of valine. Okay, just put extra CH2. So in leucine we just put extra CH2. Okay, and all the structure is similar to that of valine. Clear? Just we have to put extra CH2 in case of leucine. Okay, so this is the structure of the leucine. Okay, single letter word is L. Clear? Leucine is mainly found in where? Zippers, in leucine zippers. Okay, I hope that you study zippers in transcription, that is in molecular biology. Okay, Clear? And remember one important point about the leucine is that it is the most occurring amino acid in protein. Okay, it is the most occurring amino acid. Leucine is the most occurring amino acid in protein. Okay, most occurring amino acid is leucine in protein. Okay, clear? Next, the next structure that we will going to see is isoleucine. Okay, isoleucine. Okay, the single letter word for isoleucine is basically IL, you can say. Okay, IL. Okay, so this is a single letter word for isoleucine, IL, or you can say I. Okay, you can say I or IL. Okay, this is a single letter word. I for isoleucine and I for single letter word is also I. Okay, now just look at the structure of the isoleucine. These three are common. This is NS3 plus, this is hydrogen. Now we draw the side chain with yellow. See, just look at the structure. Here you have to put one CH like that of valine. Okay, like that of valine, and just put CH3. Clear? And just put CH2, CH3. So this is a structure of isoleucine. Okay, like that of valine, you just put this and just CH2, CH3. This is a structure of isoleucine. Clear? Very simple structure. Okay, this is a side chain. Okay, so it is also a chiral molecule. It means optically active. And the important point about the isoleucine is that the most non-polar amino acid. Okay, it is the most non-polar amino acid. Okay, so isoleucine is basically the most non-polar amino acid. Please note this point. Clear? So this is all about the isoleucine. Next, so in the next structure we will discuss about the proline. Okay, proline. Clear? So the single letter word for proline is P. That is P for proline, and P is the single letter word. Clear? Now just look at the structure of proline. Okay, in case of proline, if you look at the structure of proline, just look at the structure of proline. Okay, so in case of proline, basically proline is known as amino acid. Okay, proline is basically known as what? Proline is known as amino acid. Why proline is known as amino acid? You can find out with the help of this structure. Just look at the structure. So this is common, C alpha, CO minus, hydrogen, and this is H2N plus. Okay, now here it is the ring is formed, that is CH2, and that is CH2, and this ring will going to fuse with this, that is CH2, and this fuse with this amino group. Okay, so this side carbon diffuse with the amino group. Okay, that's why it is called amino acid. Okay, amino acid. Okay, this side carbon is basically fused with this group, is fused with this group. That's why it is called amino acid. Okay, amino acid. So this will be the structure of proline. Okay, this will be the structure of proline. Okay, so what is the function of proline? As I already told you, that proline basically disrupt the structure of alpha helix. Clear? I will explain this in our secondary structures lecture. Okay, clear? So this is about the proline. Okay, now next is methionine. Okay, next is methionine. Clear? See, this is the structure of methionine. Okay, wait, methionine. What will be the single letter word for methionine? The single letter word for methionine is M. M. Okay, that is M for methionine, M for this single letter word. Okay, just look at the structure of methionine. Very simple structure, that is C alpha, CO minus, this is hydrogen, and this is NS3. Clear? Now we draw the side chain with yellow. Just put CH2, CH2, here sulfhydryl group, and here CH3. Clear? So this is the structure of methionine. Okay, CH2, CH2, this sulfur group, this is important. Sulfur group is very important, and CH3. So this is the structure of methionine. Clear? Okay, now what is important about the methionine? The important thing about the methionine is that methionine is a sulfur containing amino acid. As you can see here, in side chain sulfur group is present. Sulfur group is present. So in side chain sulfur group is present. So methionine is one of the sulfur containing amino acid. Okay, can you name the other sulfur containing amino acid? Yes, the other sulfur containing amino acid is cysteine. Okay, is cysteine. We will look at the structure of cysteine later on. Okay, so methionine is the sulfur containing amino acid. This is the important point about the methionine. Okay, clear? And remember another point about the methionine is that methionine will not going to form the disulfide bridge. Okay, not going to form the disulfide bridge. Clear? So this is about the methionine. Okay, and single letter word is M. Clear? Next, the next amino acid is aromatic amino acid. Now two aromatic amino acids remain in non-polar side chain. Okay, so till now we discuss all the aliphatic. Till now we discuss about all the aliphatic non-polar amino acid. Okay, till now we discuss about all the aliphatic non-polar amino acid. Okay, clear? And you observed one thing, that in all the aliphatic non-polar amino acid, the single letter code for amino acid, the single letter code of the amino acid is match with the first letter word of the name of the amino acid, is match with the first letter word of amino acid. So this is the trick. For example, I just put an example. For example, glycine. In glycine, the first letter word is what? G. So its single letter code is also G. So this is a trick. In all aliphatic non-polar amino acid, the single letter code is matched with the first letter of the amino acid, first letter word of the amino acid. That is in glycine, G for glycine. So the first letter code is also G. Okay, now it will be easier for you to remember this aliphatic non-polar amino acid. Okay, now two aromatic amino acid are there. Okay, mainly out of 20 amino acid, there are 3 aromatic amino acid. Out of 20 amino acid, there are 3 aromatic amino acids. Okay, out of 20 amino acids, there are 3 aromatic amino acids, and these 3 are phenylalanine, tryptophan, and tyrosine. And out of these 2 are non-polar and tyrosine is polar. Clear? So we will discuss these 2 aromatic amino acid today. Okay, and we discuss tyrosine when we discuss the structures of the polar amino acid in our next lecture. Clear? So non-polar. So first is phenylalanine. Just look at the structure of phenylalanine. Now here is a difference came in the single letter word. That's why I told you that use the trick. So phenylalanine, the single letter word for phenylalanine is F. The single letter word for phenylalanine is F. Clear? Now how can you remember that phenylalanine single letter word is F. Okay, you pronounce like this, phenylalanine, that is Phenyl. Okay, this will not the correct because P is the correct, but you pronounce like this, Phenyl, phenylalanine. Clear? Then you will easily remember this single letter word is F for phenylalanine. Okay, clear? Now just look at the structure. Okay, simple structure, nothing complicated about these structures. Okay, this is hydrogen and just CS2 we draw with yellow, just CS2, and a aromatic ring is present, aromatic benzene ring is present. Clear? Draw with an aromatic benzene ring is present. Okay, clear? Okay, so this is the structure of phenylalanine. This is a side chain. This is the side chain of phenylalanine. Okay, an aromatic benzene ring is present in phenylalanine. Okay, clear? So now, so what is the main function of this phenylalanine? That it can absorb UV light. Okay, not only phenylalanine can absorb UV light, but tryptophan and tyrosine also absorb UV light, because this is the characteristics of this aromatic amino acid. Okay, absorption of UV light, absorption of UV light is the characteristics of aromatic amino acid. Clear? So phenylalanine can absorb UV light. Now there is a question, why phenylalanine can absorb UV light, or you can say why aromatic amino acid can absorb UV light only? Due to the pi electron delocalization. Clear? So this is the reason, pi electron delocalization is a reason that aromatic amino acid can absorb the UV light. Okay, so this is all about the phenylalanine. Clear? Now the last structure of the non-polar amino acid is tryptophan. Okay, so what will be the single letter word for tryptophan? The single letter word for tryptophan is W, somewhat different. Okay, you have to remember this, that W is for tryptophan. Okay, W is for tryptophan, you have to remember this by heart, that W is for tryptophan. Clear? Now just look at the structure of the tryptophan, then you will get understood. C alpha. Okay, just draw here, this is a hydrogen, this is a hydrogen. Okay, now this is C alpha, this is CO minus, and this is the NH3 plus. Okay, clear? Now we will going to draw the side chain of the tryptophan. Okay, the side chain of tryptophan is somewhat large. See, this is a side chain, CH2 C. Okay, double bond SC NH. Okay, this will run with this, and here is a ring. Clear? So this is the side chain of the tryptophan. Okay, so this is the structure of the tryptophan, or you can say this is a side chain of the tryptophan. Okay, clear? This is a side chain of the tryptophan. Okay, and what is the main point of the tryptophan? It can also absorb UV light. Why? Due to the aromaticity in nature, due to the pi electron delocalization of the characteristics of their amino acid. So it can also absorb UV light, and it can. And another important thing about the tryptophan, that basically tryptophan is the only aromatic amino acid that can absorb UV light at 280 nanometer. At 280 nanometer, okay, 280 nanometer. So it can absorb UV light at 280 nanometer. Okay, so this is all about the tryptophan. Okay, I hope that you understand all these structures and all these single letter word. Okay, if you have still any doubt, then you can ask me question related to this. I will try to answer all these things. Okay, clear? And remember, the name of this side chain is indole. Okay, indole. This is another important thing that I forgot. This side chain is basically indole. Okay, sometimes you may get a question on examination that match the following, and you get the tryptophan, indole, and you have to correct it that the side chain of the tryptophan is indole. Okay, side chain of the tryptophan is indole. Okay, and another thing is that the side chain of proline is known as pyrrolidine. Clear? Pyrrolidine. Okay, so these two things that I have to discuss. Clear? So the side chain of tryptophan is indole and side chain of proline is pyrrolidine. Okay, so students, that's all for today's lecture. I hope you find this lecture useful. In our next lecture we will discuss the polar amino acid structures. Okay, so till then, stay safe, stay happy, and stay healthy. Thank you.